All right, guys, how's it going? Hello, Miss Christopherson, of course, and we're going to start talking about a process known as protein synthesis, and I think it's one that you'll kind of enjoy. Kids normally do really well on this test, the DNA and protein synthesis test. So yeah, let's get rolling. Okay, let's get rolling. All right, so we got to review a couple things first and foremost. We're gonna skip this for right now, okay? Just because. All right. So let's hop on down to what is a gene, okay? So a gene is basically, think of it as being like an instruction manual for our body, okay? And basically, it's a part of DNA that's going to be coding or has the directions for a trait. Okay, you can also think of a gene as kind of like a big, huge recipe book. Okay, everybody, I'm sure your parents have recipe books at home. I know mine sit on the shelf and I don't do anything with them. How a recipe book works is you take that down, you read the recipe, you make it. Okay, and that's kind of what I want you to think about what a gene is. A gene is one specific recipe for your eye color, for your hair color, for whether or not you can roll your tongue. Actually, there are genes for enzymes inside of our body, okay? How to make those. So think of it as an instruction manual or a recipe book for basically that makes us up, okay? Now, what do I mean by a trait, okay? Now, a gene, again, is basically going to be coding for a trait. So what is a trait? It's basically a genetically determined characteristic. So the couple things that I was listing before are kind of like the hair color, the eye color, whether or not you can roll your tongue, okay? Things like that are considered to be traits, okay? Look around the room, everything that makes everybody unique, those are basically your traits. And where do you get those from? You get those from your parents, okay? So that is going to be a trait, all right? Now, where exactly do we find a gene? Okay, a gene is going to be within the DNA. All right, and how does it work? It's basically going to be the nitrogen bases that are going to spell that baby out. Okay, so what I mean by the nitrogen bases is the combination of the A, G, C, and T. Whatever combination that they're in says, hey, this is the recipe for blue eyes. Okay, which is actually pretty cool if you think about it, right? So I like this picture right here because this is the double helix, and these are all of our nitrogen bases, A, C, G, and T, and this right here is a gene. Now, our DNA is not completely made up of genes. Actually, what's cool about our DNA is the bulk of it's actually junk. We don't know what it does, okay? But we have lots of portions in there that actually do some pretty important things, okay? All right, so today we're going to talk a little bit about RNA. All right, so what is RNA? It's a close cousin to DNA. All right, what it stands for is that it stands for ribonucleic acid. All right, so RNA stands for ribonucleic acid. If you guys could just simply put down that it's made up of nucleotides, okay? And you don't have to put down contains the phosphorus, that's okay, but it's called ribonucleic acid. Remember that DNA is deoxyribonucleic acid, okay? RNA is just ribonucleic acid, all right? Now, let's go over an RNA nucleotide. Because it is a nucleic acid, which is very similar to DNA, it is going to be made up of these nucleotides, okay? So let's go over what makes up their nucleotides. All right, A, hopefully everybody remembers this one. This little guy right here is actually going to be a phosphate group, okay? So that is going to be one thing that they are definitely going to have in common. So they're going to have a phosphate. All right, we got this little pentagon piece right here. This right here is actually going to be the sugar, which specifically is going to be ribose. Okay, if you think about DNA, DNA is going to have a sugar there as well, but the sugar that DNA has is deoxyribose. Okay, so we have a phosphate, we're going to have ribose. And right here, these are going to be our nitrogen bases, okay? Again, similar to the DNA. Now, we're going to have 4 nitrogen bases for RNA, okay? And 3 of them are going to sound very familiar to you guys. The very first one is one called guanine, okay? So we have guanine, all right? We're going to have cytosine. And if you guys want to, you can just put GC, that's okay, all right? We're going to have adenine, okay, which is represented by A. All right, now this one I do want you to write out because it's a new one, and some of you guys might have noticed this in some games that you were playing or seen it surface on some quizzes or other things, is we have something called uracil. All right, please make sure that you write out the uracil, and how we abbreviate for uracil is basically just simply using a U. All right, this is the big difference. Now, really, what uracil is just kind of like a glorified thymine, okay? So it's kind of the same thing as thymine, except there is no thymine in RNA. Why? Who knows? Okay, it's just the way that it was designed. Okay. All right, so that's our RNA nucleotide. All right. Now, let's go over some types of RNA. A lot of this stuff that we're going to be talking about here might be a little bit over your head, but that's okay because in our talk today and our talk tomorrow, you're going to see some of these things resurface and maybe it might make a little bit more sense, okay? So let's talk about the types of RNA. I think you guys have a chart in your notes. The very first one is something called messenger RNA, okay? Now, we don't really call it messenger RNA. We have a cool abbreviation for it, and what that abbreviation is mRNA, all right? So messenger RNA is mRNA, okay? Now, what exactly does it do? Okay, what does it do? Is basically it's going to copy a message from a gene on DNA. Okay, it's like the messenger. What's cool, interesting about DNA is that, yeah, it's in every single one of our cells. Yeah, it's so important. Yeah, it makes us who and what we are. But it really can't do anything on its own aside from make copies of itself. If we want to express those genes, make those genes, show those genes, we actually have to go through a process that we're going to talk about today that involves this messenger RNA. So basically, we're just copying a gene on that particular strand of DNA. Okay? And like I said, this will make a little bit more sense when we hit the notes today. All right, the next one is one called transfer RNA. All right, so let's go ahead and write down transfer RNA. All right, this one looks a little bit different than messenger RNA, and hopefully you guys are looking at these pictures. All right, how we represent this one, the nickname is going to be tRNA. All right, we're going to talk about this one tomorrow. So we have our tRNA. Okay. Now, one thing I do want you guys to put somewhere up there is that basically, no, I'm not going to do that. Let's hop to the function, okay? Is that the tRNA function is actually going to be carrying amino acids to messenger RNA. All right, now probably some of you guys are like, what? Where in the world are amino acids coming from? Okay, if you look at the top of your notes, this whole process that we're talking about today is protein synthesis. And hopefully you guys remember from your biochem unit that amino acids are the monomers that make up a protein. Okay, so it's tRNA that's actually going to be bringing these amino acids where they need to be. And like I said, we're going to be talking about that tomorrow. Okay, all right, our very last one is one called ribosomal RNA. Okay, it's called ribosomal RNA, so let's write that one down. And I'm sure everybody can guess what the nickname for this guy is. We refer to it as rRNA, okay? So the nickname for that one is rRNA. Now, tricky question, where do you think we find ribosomal RNA? We're actually going to find it in the actual ribosomes that we have inside of our cells, and we got bajillions of them. What's the whole point of rRNA? Is that it's actually going to form the peptide bonds between the amino acids, okay? Again, might not make a whole lot of sense to you guys today, but we are going to be talking about this particular one tomorrow. Just kind of nice throwing it out there so it's out there. Okay, so it's going to form peptide bonds between the amino acids. Okay, and don't ask about this animation. I don't know what I was thinking when I did it. I thought it looked like a good idea. Who knows? Okay, so it's going to form peptide bonds between the amino acids. All right, so we have messenger RNA, transfer RNA, and rRNA. Today we're going to talk a little bit about messenger RNA in our process of protein synthesis, okay? All right, so with that in mind, before we hop to that, let's kind of compare and contrast what's going on with DNA and RNA, the difference between the two. They're very, very close cousins, okay? All right, so you guys have a chart in your note for that one. Put a little star beside this chart because this is actually going to be a short answer question on your test, okay? Your next test coming up, which will actually be next week, okay? All right, so DNA, hopefully you guys all remember that DNA, the double helix, is made up of 2 strands. So where it says in the chart where it says number of strands, why don't you simply just put down 2, okay? RNA is only going to be made up of 1 strand. That's it. Okay, so for RNA, just simply put down 1. You're only going to be made up of 1 strand. Okay? All right. Next one, when we talk about the sugar in DNA is one called, right up here, deoxyribose. Hopefully you guys remember that one. So DNA is going to have deoxyribose, while RNA is going to have a sugar called ribose. Okay? So deoxyribose versus ribose. That's the sugar that's going to be in those guys. All right. And last but not least, it's going to be who bonds with adenine. So in DNA, adenine is going to base pair with thymine, A and T. And hopefully you guys remember that. Okay, A and T. While in RNA, A is going to base pair with uracil. Okay, so we're going to have A and T there. And then we are going to have Sorry, I just got an email. All right. This is adenine and uracil. Okay? All right. So with that said and done, let's review our proteins. And I think you have a spot in your notes that say review proteins. Okay? Just so we remember, and we kind of talked about some of this when we were talking about the mRNA and all those things like that, is that proteins are made up of amino acids that are held together by peptide bonds. Okay? Very important thing to write down and remember. So let's jot that down. That proteins held together by are made up of, sorry about that, are made up of, I can't talk. They're made up of amino acids that held together by peptide bonds. Okay? All right. You always are building on what you've learned. Okay? That's why it's really important to remember stuff that we discussed in our notes. Okay? All right. So let's talk a little bit about protein synthesis. Now, before I show you this short video here where it says brief summary of protein synthesis, what it is, guys, synthesis means to make. So protein synthesis is where we're going to make proteins, okay? Plain and simple. We are making proteins, okay? All right. Now I have a short video clip to show you guys here, okay? Just kind of going over some of the basics, and then we'll get into the first part of that. Okay, so I need to stop it at 14:58.
So far, we have only focused on the first part of the secret of life. How, when the chromosomes divide, information is passed from one generation to the next. But the other big question is how that information is turned into flesh and blood. This is where the concept of a gene enters the story. A gene is simply a length of DNA code letters that carries the instructions to make a protein. Proteins are basic components of our bodies. They are the main ingredient of obvious structures, such as our eyes, our skin, our hair, and our muscles. Proteins also play a hidden but highly important role. As enzymes, they act as molecular machines that carry out numerous chemical operations in the cell. That helicase copying DNA at the speed of a jet engine, it's an enzyme. There are thousands of different enzymes that scurry around our cells, performing all kinds of tasks. Enzymes are the miniature engines of life. They are made of protein. So the second part of the secret of life is that the DNA code is no more and no less than a set of instructions to build proteins.
Okay? So exactly what we just talked about. So DNA is basically directions or instructions for making proteins. Okay? All right, which is what we are going to discuss right here. So, Ms. Hartsock, if you don't mind, let's give them a 2-minute stretch break. Could you hit pause on the video here? Everybody get up and stretch, stretch, stretch. And then we will talk about this process. Okay? All right, guys, so here we go. Let's get started with this. So protein synthesis is actually broken down into two main stages, okay? So do you guys notice in your notes where it says transcription? Let's put a big number 1 beside that, okay? So transcription is the very first stage in this, okay? All right, so what's the brief summary of transcription? What we're doing here is that we're making messenger RNA, plain and simple, okay? So you guys can just simply write down your notes, make messenger RNA. All right, that's the big thing there, make messenger RNA. All right, now the whole point of protein synthesis is to make proteins. All right, so we're just going to kind of get the party started here with this one. All right, where does it actually take place? The location of it is going to be inside of the nucleus. All right, so location of transcriptions inside of the nucleus. Okay. All right, so let's get rolling with this process. So right here, you guys have this little diagram in your notes, okay? Let's label a couple things, all right? First and foremost, just to kind of get you guys familiar with what we're looking at here, these lines that I have are representing the DNA backbone. So if you guys could, let's label that, the DNA backbone. The DNA backbone, again, just as a review, is going to be that sugar and that phosphate, deoxyribose and a phosphate alternating, okay? It's the part that made up the Twizzlers in your modeling activity. All right, so that's going to be your DNA backbone. All right, these guys right here are going to be your nitrogen bases. All right, plain and simple. Now, I don't include the deoxyribose and the phosphate just because it's easier to represent it like this. It takes up less space. Okay? All right, so what I'd like for you guys to do right now is actually using, this is DNA, I want to know the other side of DNA, okay? So if you could, give me the complementary side to that DNA. Now, keep in mind here, we are talking about DNA to DNA, so we're not going over any U's, okay? So for example, whoa, go back, okay? T is going to base pair with A, and C is going to base pair with G, all right? We can have T's in DNA, that's fine, all right? So you're going to have T's down here as well. All right, so why don't you guys take a few to fill in that, please? Fill her in, fill her in. All right, I'm going to go ahead and put the answer up there. Whenever you guys are done, you can take a look and check your work. Everybody get that? Okay. DNA to DNA. This is when you got to be very careful what I ask you. Okay. Very careful. DNA to DNA. Okay. All right. Sweet. Okay, so let's get this process started, okay? Now, you guys have this in your notes, and actually, if you give me 1 second, please. I should have already had this open. 1 second, please. 1 second, please. Hold on, hold on. Look at all this stuff I have. Hold on, hold on. Okay. All right, so we are right here. You just filled this in, okay? Where I'm about to talk to you guys, what I'm about to talk to you guys, but I'm about to put up on the PowerPoint right over here, okay? So right over here on this side. Everybody see what I'm circling? Woohoo, right here, okay? All right, so here we go. What you want to do is you're going to choose a gene that needs to be read or copied, okay? So kind of we'll go with that video. It's talking about how DNA also is going to have directions for making enzymes, okay? Let's say that your body needed more enzyme. Helicase, okay? So it says, hey, yo, I need some of this, so we're going to find that gene in DNA and start reading it, okay? It's kind of like finding a specific recipe in a recipe book, okay? So choose a gene that needs to be read or copied, all right? Okay. On the other side, you guys remember I showed you where to write that one? Write this one over on the other side. Okay, so write this over on the other side. Let's put down that RNA polymerase is going to attach to DNA, breaking the hydrogen bonds. Okay, the hydrogen bonds, if you guys recall from your DNA notes, is actually what's holding the T, the adenine and the thymine together. Okay, it's what's holding all of that together, keeps DNA attached. Okay, so RNA polymerase is going to attach to DNA and break apart the hydrogen bonds. Okay, again, please remember, guys, don't write things word for word. All right, even when I'm not giving you guys videos, you should be cruising through these notes. All right, nothing is different. You're doing the exact same thing that we've been doing. All right, so summarize and move on. Okay. All right, so what we're going to do is we're going to draw this in our diagram right here, okay? So if you guys could just draw that big oval, put it over the first two bases there, and let's label that as RNA polymerase, okay? So let's go ahead and draw that baby in there. Okay. All right, let's rock and roll. So what we're going to show next, and again, how we're doing this in your notes, guys, is very herky-jerky. It's hard to make it very fluid and show the movement. So hang with me. I have two animations that are going to, actually one animation that'll make it very clear for you. Okay? All right, so I just have arrows going down. You don't have to do that. Okay? All right, so here's what you guys should have in your notes right there. What I want you guys to do in the little arrow that you have underneath this one, let's write this beside that arrow, okay? Why don't you guys simply put down that DNA is split apart or separated. So write that on that little arrow going down. All right, so what we're going to do is we're going to go to it being separated. So this should be the second part of your diagram there where I show it way separated. Don't worry about labeling these again. You've already done that. okay? All right, so let's write down what's happening. All right, so where we're going to write this one is, if you could, write it off to this side, off to the right of the diagram that is split apart. Okay, so write it off to the side here. RNA polymerase is going to add matching nucleotides to the exposed DNA strand. Okay, so RNA polymerase is going to add things to an exposed DNA strand. Okay, all right, and what we're going to do is we're actually going to show that. Okay, so obviously we're going to be focusing on the side where RNA polymerase is actually going to be attached. Okay. All right, so let's do this. So now here, what you guys got to realize is that we got RNA polymerase. We're making RNA here, okay? So we're making RNA. So this is when we go back to our new rules that you guys just learned. So cytosine and guanine, C and G come together, and A and U come together. Okay, A and U come together. So right here, when kids see this T, they're like, what? Who goes with that one? Well, if I have a T in DNA, who could base pair with that in RNA? Who likes T? Okay, and that's going to be adenine. So let's go ahead and put an A right there. And actually, guys, you can draw on your RNA polymerase again down there. I forgot to tell you to do that. Okay, so go ahead and draw on your RNA polymerase down there. Okay, oh, I already gave that one away. Okay, so we got the A right here, and then the U is going to come in. Okay, now what I want you guys to do is I want you guys to finish it. Give me the rest of them. Okay, so my baby's crying. We got finish the rest, please. Okay, so C would be G, and just continue on down. Okay. All right. So what you guys should have here is this. And if you notice, Miss Chris made a mistake somewhere, right? Oh my God, teachers make mistakes. Right down here, you guys notice I put a T. I screwed up, and it's hard to fix because I'll show you the slide when I'm all done with it, okay? That should be a U, all right? So check your work. Everybody feel pretty good about this? Once you get the hang of it, it's actually really easy, okay? It's just you got to really pay attention if I'm asking for DNA to DNA or DNA to RNA. Okay, here's definitely DNA to RNA. Okay? All right. So if you guys could, underneath your strand, just put messenger RNA underneath that so you know that that's what you're making. Okay? If you recall, the whole point of transcription is to make messenger RNA. Okay? All right. So the RNA polymerase is all the way down. Okay? And RNA polymerase is going to leave. All right? At this point, guys, I don't care where you write it. It's up to you. Okay? RNA polymerase is going to leave. Okay, so there you go. I showed you it leaving. It says peace out. Okay, peace out. All right, what's going to happen next is that mRNA is going to leave the nucleus. Okay, again, you can write that somewhere. Maybe you can put all this over here. Doesn't matter. I don't care where. So mRNA is going to leave the nucleus. Okay, so you're going to see this red strand disappear. Poof. All right. And my DNA is going to come back together. Now, please note, guys, I only copied one side of DNA because RNA is only a single strand, okay? Remember, in our comparison of DNA and RNA, we're only going to ever copy one side of DNA and making RNA, okay? All right, so my DNA says, hey, I'm done here. The DNA is going to recombine, meaning that it's going to come back together, okay? So the DNA is going to come back together, or you guys can put down recombine, okay? And then that's that, all right? That's that. So what you guys have at the bottom of your notes is you actually have this, and that is basically going to be transcription. Okay, kind of crazy stuff. Now I know the notes don't make it very clear, okay, so let's actually look at an animation together to help you guys out, okay? All right, so here what I have is I got my DNA, all right? So you're going to see RNA polymerase attached to it, and actually, guys, it's way more complex than what I'm telling you here, okay? Kind of like the DNA replication business, but you get the general idea, okay? So RNA polymerase is going to come and attach, okay? It's a big old blob, bloop, okay? As it moves down, it's unzipping the DNA, and this is actually your mRNA that we're making. How I showed it is I showed the DNA splitting completely apart. It actually doesn't do that. It's just hard for me to show it like this on a PowerPoint. It only does it a little bit at a time, which is pretty cool. Okay, comes back together. RNA polymerase says peace out. And if we were to watch that, this part right here, you'd see it leave. Okay, I'm actually going to show it to you one more time. RNA polymerase is going to attach. Again, it's a much easier visualization than your notes. Breaking those hydrogen bonds, adding RNA nucleotides to create messenger RNA. Notice the U's, okay? When it's all done, RNA polymerase leaves, DNA comes back together, and this will eventually leave the nucleus, okay? Piece of cake, right? All right, let's watch one more thing about it, okay? One more thing. We're going to hop back to this video right here, okay? Oh, it's unfortunate they're showing North Carolina. Wah, wah. Okay, let's talk about right here. Okay, so this is actually going to show you what it looks like inside of our cells. So this is actually happening inside of the trillions of cells in our body right now as we speak. Okay? All right, here we go.
Within 20 years. What you are about to see is DNA's most extraordinary secret. The innermost workings of how a simple code is turned into flesh and blood. This is what Francis Crick called the central dogma of modern biology: how DNA makes protein. It starts with a bundle of factors assembling at the start of a gene. It's these that trigger the first phase of the process, reading off the information that will be needed to make the protein. The gene is the length of DNA stretching away to the left. Everything's ready to roll. 3, 2, 1.
Check that out. Isn't that cool?
The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the two strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA.
Where's my cursor? All right, so this yellow stuff right here, this is actually going to be messenger RNA. This is the DNA that we're unzipping a little bit at a time. All these specks are actually the nucleotides, and this right here is RNA polymerase, okay?
The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to make an exact copy of the A's, C's, G's, and T's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related nucleic acid known as U. You are watching this process, called transcription, in real time. It's happening right now in almost every cell in your body.
So cool.
It's a bit like taking the information off a hard disk on a computer and put it into memory, making it a real program that's running. So that process of DNA to RNA is like running a program, double clicking on an icon.
When the RNA copy is complete, it snakes.
Okay. So this is where we left off, is that we made messenger RNA. We are obviously going to do more with that, but we are not going to get into that today. I just think that is the coolest thing ever. What can I say? All right, so let's look at I'm sorry that's messed up in your notes, guys. All right, let's take a look at this thing right down here at the bottom of your notes, please, okay? Where it says practice, okay? So what I've given you here is I've given you a strand of DNA, and what you might see them ask on the EOC or me ask on a test or a quiz is I might say, all right, transcribe this piece of DNA, okay? What that basically means is that I want you to give me the messenger RNA for that, okay? So, for example, I'll give you this first one right up here. I have TAC, so I'm making messenger RNA here, not DNA, all right? So, in RNA, who base pairs with T, that would be A. A likes U, and C likes G, okay? So, we have A equals U, and G equals C, all right? So, why don't you guys finish that for me? Continue on, cruise on through. Okay. All right, when you guys are done, take a look up here and check your answer. Okay, like I said, once you get the hang of this, it's actually kind of fun because it's a lot easier than some of the crazy stuff that we've been doing. Like, it's easier than the cell cycle, definitely easier than photosynthesis and cell respiration. Like I said, it's once you get the hang of it. Okay? Hopefully everybody's cool on that. Okay. All right. So, with that said and done, what you guys are going to be working on today is that you guys are going to start an activity called Alien Encounter. It's actually really fun. But, you know, I think all this stuff is fun. All right. So, with the Alien Encounter, it is right here. And Miss Hartsock has the directions for this, so she's going to help set you guys up for that. But if not, the directions, or if you're confused still, the directions are right up here, okay? Basically, all that you guys are going to give me today is you have a chart, a sheet that looks very similar to this. And what we're doing here is that, I guess I should read this top part right up there, is you made a discovery, an archaeological discovery, and you find alien remains, and you're basically trying to figure out what they look like, okay? So each group is actually going to get a sheet of paper that looks very similar to this, and these are all basically the genes in that particular alien species, okay? So what you're going to do with these genes right here, okay, gene A, blah, 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 on this chart, you're actually going to write down where it says DNA, that's where you're going to put that code right there, okay? So each of these, gene A, B, C, this part right there, you're going to write by the DNA, okay? For the messenger RNA, basically, this is all that I want you guys to do for today, is to give me the messenger RNA. So, for example, just do exactly what you did in that PowerPoint, okay? Or, like I said, transcribe. So put it into the RNA, and then tomorrow we will actually make a protein from it, okay? And we'll get an idea of what those look like. All right, so you're writing down the DNA today. You're going to write down their messenger RNA, and then you stop, okay? Make sure you put down the alien number. All right, don't worry about naming it just yet, okay? And actually, this sheet with the directions, you guys are going to leave in the basket, and you are going to leave these in the basket as well. Do not take those with you. Now, you guys, I'm not quite sure if she's going to give you each one individually or she's going to put you as teams. If you guys are in groups of 2, I still want you guys to spend the time giving me the messenger RNA because it's practice, okay? And that's the whole point of these activities, is to practice the notes. The more you do this, guys, the quicker that you're going to be able to power through a lot of this, okay? All right, once you get done with that, what you guys can work on is you can work on your homework, okay? Tells you what numbers to do up there on the wiki. And then if you get done with that, what you guys can do is I link some review games for the cell cycle, all right, where you can just continue to practice with that. Okay, sound good? All right, guys. Like I said, I hope all is well, and I will talk to you tomorrow. You're getting an awful lot of videos, and that is just because these topics are really hard. They're just hard. So you're not always going to have videos, kind of like what you saw yesterday, and you're not going to have a video on Friday either. Okay? All right, anyway, guys, you take care and keep being smart and behaving yourselves. All right, bye-bye.